So we have covered fertilization, and we have seen that what we have right now is a cell, a single cell, we are calling the zygote. This single cell called the zygote is within the fallopian tube. We want to see what happens to this zygote during the first 7 days of life. That's what we mean by the first week of development. If you remember our story, we've talked of the events that take place before conception, and this is gametogenesis and fertilization. We are done with that. After conception, we divided this period into 3. We said the first 2 weeks is what we are calling the pre-embryonic period. Week 3 to week 8 is what we are calling the embryonic period. And week 9 until birth is called the fetal period. So we are now here. We will talk about week 1 today. Next week, we'll talk about week 2. What we need to learn in the first week of development are the following. We are going to explain the events that occur during the first week of development. We are also going to look at the morphological changes that take place during this first week of development, in the understanding that we can't be talking about the zygote forever. The first week of development ends by formation of a structure called the blastocyst. We are going to see the parts of the blastocyst. So, straightforward, the first week of development occurs largely within the fallopian tube. The reason why that is so is because actually it is within the fallopian tube that the oocyte was formed. And the oocyte is not like sperm. The oocyte is relatively composed, moves slowly, not like sperm na kierere zake. So if the fertilization took place within here, the ampulla of the fallopian tube, then this thing need to move from here to there, from the site of fertilization to the site of implantation. That movement is very slow, so that the first 4 to 5 days, the conceptors will actually be within the fallopian tube. And perhaps the latter days of the first week, the ones where the conceptors will be in the endometrial cavity. So what happens to the conceptors during the first week? The conceptors will be propelled from the site of fertilization, which is the fallopian tube, to the site of implantation, which is the endometrial cavity. Key to this propulsion would be the ciliary activity of the fallopian tube. And that is why if this cilia is not functioning properly, a woman can be subject to ectopic pregnancies. The oocyte never reaches, or rather the conceptors will not be able to reach the endometrial cavity. Apart from movement of the conceptors from the site of fertilization to the site of implantation, we agreed that one of the things that fertilization unlocks is that ability to undergo mitosis again. And sure enough, the zygote will begin to undergo mitosis. The mitotic division of the zygote is what we call cleavage. So you can imagine if these cells are actually undergoing cell division, it means that there'll be some morphological changes which occur on these cells. Let's look at these morphological changes. On the first day of development, the sperm entered the oocyte. When the sperm entered the oocyte, it made the oocyte to complete its second meiotic division so that we form the mature ovum as well as the second polar body. So the cell you're seeing here has two polar bodies. It means that it has completed the second meiotic division. However, the male pronucleus has not yet fused with the female pronucleus. So you have a cell with two pronuclei. We called it the otid stage of development. From this otid stage of development, the two pronuclei are going to fuse in such a manner so that eventually we are going to have a single cell with a single nucleus that is now diploid. And that single cell with a single nucleus is what we call the zygote. So on the first day, we have the zygote. Development begins with the zygote. The zygote is still covered by the zona pellucida. As a matter of fact, the zona pellucida has hardened. The hardening of the zona pellucida prevented polyspermy. And you can see, even in this image, the number of sperms which have been trapped through the hardening of the zona pellucida. So you have a zygote with an intact zona pellucida. We agreed that the zygote begin to undergo mitotic cell division. The series of mitosis that the zygote undergoes is what we've called cleavage. Now, let's consider a single event of mitosis, a single cleavage event. A single cleavage event will give us two daughter cells. These two daughter cells are still enclosed within the zona pellucida. These two daughter cells are actually seen usually either within the same day or the following day. We call this stage of development the two-cell stage. These two cells will continue to divide. So it means they still undergo cleavage. Now, let's assume that both of them have divided. So it means that you have something with four cells enclosed within the zona pellucida. This four-cell stage is usually seen on the second day of development. So remember, on the first day, we had the zygote. Zygote begins to divide so that you can have the two cells on the same day or the following day. Then those two cells also divide. Still on the second day, we have a structure with four cells. We call that the four-cell stage. Want to give you another question based on those four cells. And I want you to choose from one of those. I'll give it as a poll. That's the last poll you're doing. Remember, I told you I combine these polls and they'll contribute your assignment marks. I'll not give you a separate assignment for marking. All right. You can respond. All right, we're good now. Only one person has not responded. So this one now, most of you have gotten it wrong. When we say a cell is totipotent, it means it can form everything. When we say a cell is multipotent, it means it can form many things, but not everything. If we say a cell is differentiated stem cell, it means that it can give rise to a particular lineage of cells. For example, osteoprogenitor cells. They are differentiated stem cells. They are stem cells for bone. When you say a cell is terminally differentiated, it means that it cannot give rise to any other type of cell. It's the end stage for it. Now, with that understanding, the 4 cell stage are actually totipotent cells. Because from these 4 cells, you're going to get the brain, you're going to get bone, you're going to get hair, you're going to get even the placenta. They have total potency. A good example of multipotent cells would be hematopoietic stem cells. Hematopoietic stem cells are multipotent because they can give rise to different cell types of blood. Cells of early embryonic period are totipotent cells. A closer term for that is pluripotency. But pluripotency is somewhere between totipotency and multipotency. I removed it so that you don't have that ambiguity. Now we are saying we have a cell, a structure with 4 cells. We have called that structure the 4 cell stage. And we are on the 2nd day of development. Definitely, cell division continues. And that means that we add to the number of these cells. Perhaps not that the cells are actually dividing at the same rate. So I don't want you to fix your mind on now 4 cells give us 8, 8 will give us 16, then 32, then 64. It is still possible now that there are some cells which are slower than others. And so it's still possible to get a mass with 20 cells, a mass with 31 cells, a mass with 40 cells. They don't have to be the squares of 2. With that in mind, therefore, there reaches a time where you have a mass of cells that contain between 12 to 32 cells. That period where you have a structure that contains between 12 to 32 cells is noted on the 3rd and the 4th day of development. This structure is called the morula. There are 2 things I want you to note between this and this. I want you to notice that the zona pellucida is still intact in both of them. But I also want you to note that the cells here are fewer than the cells here. Now I want you to think, if the zona pellucida is still intact in both of them, yet the number of cells here are more than the number of cells here. it can only mean two things. One is that the cells here can only be smaller than these cells. And sure enough, they are. But there's another thing. It can also mean that the cells here are too squeezed compared to these ones. And sure enough, they also squeezed. The process of squeezing of the cells of early development is known as compaction. The cells of the morula are compacted. This compaction is believed to be important for intercellular communications. These cells communicate with each other. They identify each other with these communications. So please note that the morula has more cells. That's true, because the cleavage has been going on. But the zona pellucida is still intact. And because the zona pellucida is not expanding and is limiting, if you're increasing the number of cells within the zona pellucida, then it can only mean that those cells are smaller. And true enough, the cells of early development increase in number with every cleavage process, but decrease in size with every cleavage process. The size of the individual cells decrease with every cleavage event, but the number of cells increase with every cleavage event. As the number of cells increase and the zona pellucida is intact, the pressure within the zona pellucida is high, and that's what we're calling compaction. The cells are squeezed. Now let's bring this morula to the next page. So it's here. We have the morula between the 3rd and the 4th day. Here we have something between 12 to 32 cells. What happens to the morula? The morula will continue, the cells of the morula will continue to divide. That's cleavage. That means that the pressure within the morula will continue to increase. That is compaction. So compaction is going on, cleavage is going on. To add to that, something drastic also happens. A cavity forms within the morula. This cavity appears on the 5th day. This cavity contains some water. When that cavity is present, we do not call it the morula anymore. We call it the blastocyst. So on the 5th day, we have a structure we are calling the blastocyst. Also note, the zona pellucida is still intact at this stage. Let's bring this blastocyst to the next page. This type of blastocyst exists on the 5th up to the 6th day. That type of blastocyst. As cell division continues, which is cleavage, the size of that cavity also increasing, it means that the pressure within the blastocyst is extremely high. The compaction is at its maximum. And what would happen if the pressure is too much from within? The zona pellucida has no choice but to give way. And sure enough, the zona pellucida gives way. If this was to happen to your eggs at home, then you'll call it hatching, where those chicks come out. Interestingly, we still call it hatching here. The zona pellucida gives way in a process called hatching. And when the zona pellucida gives way, I want you to look here. This is where the zona pellucida gives way. This region of the zona pellucida that's next to the cavity. This region of the zona pellucida is the one that gives way. We call this region the embryonic pole, as opposed to this side, which is called the embryonic pole. The side with the cavity is called the embryonic pole. And I'll explain to you shortly why it's called embryonic and why this one is called embryonic pole. So the zona pellucida gives way at the embryonic pole. And what happens next is that the blastocyst will come out through that defect. As you can see here, this is a small blastocyst, but it was fitting this whole mass. But there's a break there, so this whole mass is coming out in a process called hatching. Once the blastocyst has undergone hatching, it will then look like this. This structure, we will have it from the 6th day up to the 8th day. We still call it the blastocyst, actually. But to distinguish this type of blastocyst from this type of blastocyst, we call this one the early blastocyst, and we call this one the late blastocyst. This is early blastocyst. This is late blastocyst. I want to ask you a question which you are going to discuss in your breakout rooms. The question is this. I want you to state the differences between the early blastocyst and the late blastocyst. So look at them. You can even take a photo of it, because when I divide it into your groups, you're not going to see them. I want you to talk about the anatomical differences that you can see between the early blastocyst and the late blastocyst. So that question, you need to do it in 2 minutes. Let me create the groups for you. Okay, the groups are already created, but I'm yet to open it for you. Okay, I've opened it now for you. So join your group. You have 2 minutes for that question. Right, welcome back. So I want to hear from maybe 2 or 3 people the differences that you talked about in your group. You can unmute yourself, then say something. Anyone, feel free.
Can you say that in the Sangama? Can you say that in the early blastocyst, it is covered by the zona pellucida, while for the late one, it is not covered by the zona pellucida?
Absolutely, that's right. Anything else?
Early blastocyst has narrower cavity, while late blastocyst has wider cavity.
That's accurate. Good. Anyone else?
Pressure difference.
Sorry?
Pressure difference between the two.
You can see pressure.
Let's see. Oh, we talk about morphological cell.
Yeah, we want morphological differences. But I agree with you, the pressure in the early one is higher than the pressure in the later one, except that that one is not anatomical. Any other difference?
The cross-section of the late blastocyst is bigger than that of the early.
Oh, you mean the size?
Yes.
Okay, good. So there's a size difference. Any other difference? Someone to comment on the shape of the outer cells. Anyone?
They are stretched.
Yes, there's a better term. Which other term do you want to use? Instead of saying stretched.
Squamous.
Very good. So the late blastocyst, the outer cells are squamous. And the early one, the outer cells are? What term do you want to use for those cells? Okay, ni kama mtaniambia siku ingine, haina shida.
Cuboidal.
Okay, that's better. All right. Let's talk about parts of the blastocyst. The individual cells of the blastocyst are called blastomeres. The term blastomeres is not really limited to the cells of the blastocyst only. It can also apply to the cells of the morula, the cells of the 4-cell stage as well. Blastomeres. The blastomeres of the blastocyst exist in 2 different populations. There's a population of cells which are on the inside of the blastocyst. We call that the inner cell mass. The inner cell mass represent the population of cells that will later become the baby. So it means that they're the ones that are going to become the embryo. For that reason, we call it the embryoblast. The term blast means immature or forming. The same way we talked about the fibroblasts, osteoblasts, it means they are forming or immature. The population of cells of the blastomere, which are on the outer side, you want to call them the outer cell mass. This outer cell mass represent the population of cells that will form the placenta. So we call it the trophoblast. The term trophoblast means, okay, the term trophic means to support. And so this is the supporting layer, the layer that will become the placenta. Remember, placenta is a supporting layer. This cavity within the blastocyst is called blastocyst cavity. With reference to the trophoblast layer, there are 2 poles of the trophoblast that we can describe. There's this side of the trophoblast that contain the inner cell mass, or that is next to the inner cell mass. And there's this side of the trophoblast that is away from the inner cell mass. The part of the trophoblast away from the inner cell mass is called the embryonic pole. Now you know why it's called embryonic pole. The embryonic pole is the side that is used during hatching. So this side of the trophoblast adjacent to the inner cell mass is therefore called the embryonic pole. And this is the side that the blastocyst is going to use for implantation. The embryonic pole is used for implantation. Embryonic pole is used for hatching. Right. Evening passed, morning came. That was the first week of embryonic development. Let's make a summary of what happens during the first week of development. We have said during the first week of development, the conceptus moves from the site of fertilization, which is the ampulla of the fallopian tube, to the site of implantation, which should be the endometrial cavity. The main mechanism that facilitates this movement is the ciliary activity of the fallopian tube. This is also assisted by the contractions of the fallopian tube, especially for it to pass through the isthmic portion of the fallopian tube. We've agreed that the first few days, therefore, the conceptors will be in the fallopian tube, then the latter days in the endometrial cavity. As this movement takes place, these continuous morphological changes. These morphological changes are being caused by that continuous cell division, which you call cleavage. Also by the fact that the cells are squeezing amongst themselves, causing a lot of pressure within, and therefore the zona pellucida gives way in a process we are calling hatching. Importantly, if you look at the morphological stages, we have the zygote, which is a single cell seen on the first day, then 2-cell stage, which is seen either on the first or the second day, 4-cell stage, which is seen on the second day, the Morula stage, which is 12 to 32 cell stage, seen between the third and the fourth day, and thereafter the blastocyst stage, which is seen between the 5th day, actually up to the 8th day of development, except that on the first day, that day 5, we talk about the early blastocyst, and later on the late blastocyst. And you've stated the differences between the two. Remember, the blastocyst continues into the beginning of the second week, because we're talking about it entering day 8. Importantly, the first week ends when 2 populations of cells have been formed. The population of cells that will become the baby, and the population of cells that will constitute the placenta. So therefore, we can say that the first week of development is a period when we actually establish what will be embryonic, that's the inner cell mass, and what will be placental, that's the outer cell mass. Remember, the placenta is not embryonic. It is not the baby. And that's why when we're talking about molar pregnancy is not a pregnancy that's going to give you a baby. It's just placental tissue. Such pregnancies must be terminated. At the end of the first week of development, the conceptus is ready for implantation. Remember, the structure that's going to implant is now the blastocyst, the late blastocyst. It's the one that's going to implant. So that is the first week of development. We will stop there now. If you have questions, you can ask. Let me see if there are questions in the chat, but feel free also to unmute yourself and ask a question.